85 slides, got it. 85 slides into, I don't even know. Anyway, I'll do my best. It's probably not going to flow too smoothly, but my graduate student, Dr. Yusuf Moti, who's matriculated, myself and Dr. Keating, I had come up with this idea that, well, I've been teaching biology cancer for about 45 years.
Hey,
Been at FAU for 50.
just something you can take if you want. Just sign your name. Just free cookies and stuff.
So a little background noise. Do we need, Michael, do I need a mute background noise or?
They can mute. Just have everybody mute, please.
Just everybody mute.
Yeah.
So, yeah, having taught biology cancer that long and having read about all the various means by which cancer cells evade both the immune system and chemotherapeutic approaches, I came up with 3 compounds that I thought number 1 criteria is that they could not be DNA poisons. They could not be mutagenic, because as an immunologist, I know that DNA poisons are very, very immunosuppressive. And what I wanted to do is not only kill the cancer cells, but have them die a very special death. It's called immunogenic cell death. Very well characterized, very well known. And when they die an immunogenic cell death, the immune system is alerted to come take a look, process antigens, and then activate cytotoxic T cells. So the title reads targeting breast cancer, but we also tested melanoma, leukemia, and we tested breast cancer of both humans and mice. And I'm sure you're aware of the problems with current therapy. It is toxic to normal cells. So that was one of my main criteria. And I think I'll present evidence that we found a means to selectively kill cancer cells and not affect normal cells. And therefore, we think it's pretty specific. And most conventional chemotherapies, they give you it's about as ridiculous as saying using a single antibiotic can tell you to go home, take 1 antibiotic. Chances are you become resistant. So we wanted to develop something that my doctoral student, when it worked beyond our wildest expectations, he called it the trifecta. I call it oncotriad because I wanted to include the oncogene or the oncology aspect of it. So yeah, just cutting right to the chase, we tested both a mouse breast cancer cell line, a human breast cancer cell line, and you can see this is what's called a colony forming assay. So you can't fudge this data. I mean, I guess you could somehow, but seeing is believing. So these are the colonies that are produced by the mouse cells at I think we see them at 200 or 300 cells per plate, and you leave them undisturbed in the incubator. And you do the same thing with the human breast cancer cells. And so the 3 compounds, boy, they annihilated them. But wow, if it's that effective, might it be toxic toward normal cells? And so what I told my doctoral student, I said, well, let's pull out some, we have lots of human PBMCs, human peripheral blood, frozen in liquid nitrogen. So we pulled those out. And I said, well, and I should have pointed out that, well, when it says low dose, here's what's really amazing about this, because I took out lots of slides. We first determined the ED50. And then when we mix these 3 compounds together, we found that we could use them at 6 times below their effective dose, which is pretty remarkable. And so, wow, what happens when we use them at not 6 times below their ED50, but we even use them at 3 times above their ED50 to see whether or not they would have any adverse effects on the viability. So we're measuring viability untreated PBMCs, human white blood cells. And here's the EC50. Obviously, well, I shouldn't say obviously, we're pretty amazed to see that there was no decrease in viability, but then we kicked it up to 3 times above their EC50, combining them 3 times above. And you say, well, surely we're going to reach a point where they're going to be toxic, but they're targeting 3 different signaling pathways and very, very effective at, so I think is pretty selective killing. Now, one of the things about cancer cells is they're dividing rapidly. I'm trying to watch this time here. So the first experiments we did were thawing out the peripheral blood mononuclear cells, putting them in culture medium, and then treating them, really whacking them 3 times above with this combination. But I told my graduate students, cancer cells are rapidly dividing, we're killing them. Maybe it's because the immune cells aren't dividing. So if we kick them into division, maybe our magical 3 compound combination will be toxic, but nope. At the EC50, still no toxicity. So phytohemagglutinin is a well-established means to trigger division of white blood cells. And as I said, so we have what appears to be selectivity, non-toxic toward human whole blood-derived peripheral blood mononuclear cells. And then I said, well, wait a minute. So we kicked them into division, but cancer cells are in a hypoxic environment. And so maybe the immune cells, when they come in there, they'll be in a hypoxic environment, but it's also non-toxic when we take the human white blood cells and we put them under hypoxic condition or low oxygen condition. So this is another graphical representation of the here in this graph, viability. I don't know what cell line this was. No, it's not a cell line. It's again testing the human PBNCs or normal healthy white blood cells. And what we also did was, well, let's compare this. Maybe something's wrong here. Let's use a known cancer chemotherapy compound, doxorubicin, and we're not going to use it at 3 times above their EC50. We're going to use it at their use. This is what they inject. This is the blood concentration you'd find in a breast cancer patient. And boy, just as one expects, it knocked the viability in half. And what's even more amazing is doxorubicin is cardiotoxic. Very known to be highly cardiotoxic, and that's understandable. It is a very toxic compound. So I'm moving along here. Too many slides. Again, this just reiterates what we can measure a number of different ways. So I had all kinds of flow cytometry data. We run flow cytometry in the lab, and so annexin 5-7-AAD that indicates apoptosis. And again, you can see, boy, doxorubicin kills like crazy. Well, it causes the apoptotic cell death and eventual necrosis at 35%. Whereas our compound, our magical 3 compounds here, is roughly equivalent to untreated. So again, we're showing it's selective and relatively non-toxic for human white blood cells. And this is just another, again, we treated them, stimulated them to make sure that, if they're dividing, maybe it's going to be toxic, but you can see that doxo knocks them out again, and ours doesn't. And this is a number of, so not only we do flow, we did a number of other tests, metabolic tests. He actually sat there and counted the microscope cells per ml. And so he's looking at the untreated, ours And then if he counts the cells, you still have cells around. They haven't undergone apoptosis, so you don't see this lighting up on the flow cytometer, this 45%. So the cells are at this juncture, I forget how many hours this was, they're still hanging around, but they're on their way out. So the real exciting part of the compounds that we're working with is a brand new concept in the field of immunology. You know, you've probably heard about immunotherapy. Well, you can induce an immunotherapeutic response within a patient, depending on how you kill the cancer cells. So this is a well-established, I mean, there's a publication that must have 500 authors on it. You know, how do you prove that your regimen is killing and eliciting an immunogenic cell death that will attract the immune cells to the point of death, cell death of your cancer compound, and will activate them. So there are three major hallmarks, and those are calreticulin surface expression. The cell's dying. If it's dying in immunogenic cell death, it'll kick out calreticulin on the surface. It'll release ATP. ATP should only be inside a cell. It's a danger signal when it's out. And this one, HMGB1, should only be associated with DNA. And when the immune cells, they've got receptors for these. The innate immune cells have receptors. And so they pick these up and they'll home to the site of the tumor in 15 minutes. So catch my breath here. So I guess we're just emphasizing again that this is annexin 5, 7-AAD, and a flow cytometer. When you see those two markers, you know that cells are dying, and untreated 8 hours, 12 hours, and again, cells per hour out. But the important thing is, again, we're adding them at 6 times below their individual effective dose 50. So against these MCF cells, they're the human breast cancer cells. And how come this doesn't want to go? I don't know why I didn't show you the whole. Okay, so yeah, I should be clicking on here. I wanted to move too quickly. So just to sum up, we had to get data to prove that our cells were dying immunogenic cell death. So here's the literature. You look at all the literature, the average of what you should see in terms of ATP, 450. What did we measure? ATP after the trifecta therapy, 3 compounds, 709. Plenty of ATP coming out of those cells as a danger signal. HMGB1 should be in DNA, 150 nanograms per ml. Wow, 187 nanograms per ml. And we could show you the range and statistic, but we just kind of summarized everything here. The percent cell death that induced immunogenic cell death in MCF7 was 40%, and the trifecta. Oh, from the literature, other single compounds that induce cell death would kill at about an illicit immunogenic cell death in about 40% of the population, whereas our trifecta about the same. And that might be, so this is, how am I doing on time? I'll quickly, without going into any more detail here, I'll show you. So we're treating cells in a monolayer, but one could argue that, well, that's a phony system. So the newest way to treat cancer cells is to form tumor spheres, because cancer cells don't grow as a single monolayer in the body, they grow 3-dimensionally. So we were able to follow the literature and produce these things called tumor spheres. And when we take the tumor spheres and we treat them for 48 hours with our combination, you can see the uptake of trypan blue. not here. You see a background of trypan blue, but it's not taken up and concentrated in the cells. So trypan blue is a
The only
well-established
one
dye.
that's
Come again? It is a well-established dye. If a cell dies, it will take up trypan blue and stain blue. So that's pretty graphic evidence. And we also extracted the dye and got, I think there's some quantitative data. Here's our 3 compounds, untreated, and viability as a percentage based upon that dye, the trypan blue dye. And what we were kind of surprised, we thought, well, okay, let's test the old cardiotoxic doxorubicin. That is the go-to drug for breast cancer. And look what it does. Look what it doesn't even kill the cells in the tumor sphere, which is a model. So all the previous work on doxorubicin is done in a model layer. Oh, wow, it's toxic. Oh, let's take that and use it on patients. But if you look at now the most accepted model tumor spheres, you don't see much effect at all. So that probably explains why doxorubicin as a single agent, you see a lot of tumor cell resistance. Now, this 60 micromolar is even higher than 25 micromolar is the usual blood level. So this is even higher, and we're not killing those cells in the tumor spheres. And what you find is that when cells are in a 3-dimensional matrix, it's protected. Oftentimes we'll secrete a fibrous matrix around the tumor sphere, and doxorubicin didn't do the job on them. And whether it's mouse or whether it's human, we were amazed that the conventional chemotherapy treatment, we thought, well, it's toxic, it would just annihilate them, better than ours maybe. But so the conclusions: we have a novel formulation. They're non-mutagenic compounds. They don't interact with DNA. Highly effective in combination. Well below their individual clinical dose. Only one of them has been approved by the FDA for clinical use, and that is methotrexate. Methotrexate's not a mutagen. It robs the cancer cell of folate. And 2-deoxyglucose, lost my mouse here. So 2-deoxyglucose is definitely, it's an analog of glucose. It's not mutagenic, but boy, it robs the cancer cell of sugar, and cancer cells are addicted to sugar. They exhibit something called a Warburg effect. They suck in sugar at about 100 times the rate of a normal cell. So yeah, you would have maybe some effects on a normal cell, but it's not going to kill them, as we saw with our healthy PBMCs, even when they're stimulated to divide. So and wagonin is a flavonoid that does a number of different things. And I did have a really nice complicated slide, but I didn't think I'd have time to go through all the complications of this slide to show you the multiple interactions of these 3 compounds and the pathways they block and stop. So that's it. It's effective against tumor cells and doesn't affect or target healthy immune cells. And this is really exciting. I showed this data to an oncologist here in town, Dr. Sam Reddy, and he really lit up when he saw this, because now there are a lot of monoclonal antibodies that are effective against neutralizing some of the blocking molecules that cancer cells have. But they're pretty effective in melanoma, but I don't think they would hold up in comparison to what we have. And more importantly, it's effective against 3D tumor models. And the literature states that 3D tumor models are really nearly equivalent to what you're going to see in a mouse. So we haven't gone to mouse studies. We filed a patent. The university declined. I disclosed the patent on this. The university declined, probably because of shortage of funds during the pandemic. And so we filed a patent ourselves. It's patent pending. So now we can share it with you, and hopefully it'll be a completed patent. And that is the conclusion of this talk. I did have another talk if some of the students want to hang around later. The current research I'm doing in my lab now is on endometriosis, which affects 1 in 10 women throughout the world. And so I've got some really good ideas, a double whammy there, instead of, most of the time it's one drug and doesn't work. So anybody want to ask any oh, I see chat.
I was going to say, Dr. Hartmann, hopefully then next year we can talk about my compounds too.
Yes.
Including one of mine actually wipes out endometriosis cells, but we're not going to talk about that just yet. Michael and I found that out.
Okay, yeah, so next year we'll have a whole endometriosis presentation, and we'll compare your data with my approach. And then we'll also, but I'm going to get after Jay to make sure he tests normal cells, not just the endometrial cells, because he's testing marine compounds, and most marine compounds are highly toxic because they want to ward off predators or infectious organisms in this soup in the ocean. So that's why in the study I presented, what I think is, I think it almost borders on scientific fraud for somebody to claim they have an anti-cancer compound. And there are lots of publications from pretty prestigious places that have a new anti-cancer compound. And you could dig through the whole paper and they never tested it against normal cells. If it's equally toxic against normal cells, you're going to kill the person. You're going to wipe out their immune system and they'll die of an infection. And they won't have any immune system to clear out those few remaining cancer cells.
Can
Jay, that
research
sounds like a
lab
challenge
full
from
for the summer.
Dr. Hartmann himself.
So I see, so GM, I don't know who that is, but GM, send me an email. It says, is your research lab full for the summer? So send me an email and with your background. And it's just J Hartmann with one N, JHARTMAN at FAU.edu. And send me a check for $10,000 for the research. No, I'm kidding. We need the endometriosis. We need to get some funding. We had very sizable funding working on this project.
I ask a question?
Sure, Larry.
So why is this immunogenic cell death? What does this have to do with the immune system?
Well, when a cell dies, it can die very quiescently and not alert the immune system. So if it's a cancer cell just dying, a apoptotic or necrotic cell death, but not an immunogenic cell death, it won't attract any immune cells. So immune cells, in order to get an immune response, you first have to attract cells. They're called innate immune cells, dendritic cells, and macrophage. You have to first attract them to the site of the dying tumor cells. So when they get there, after some of these compounds that we talked about, the three, they're chemotactic. They attract the macrophage and dendritic cells. And when they get there, they're going to see dying cell debris, and they have scavenger receptors. So they're going to take all this in. And when they take it in, if it's sensed as danger, and ATP is a danger molecule, so they're going to be picking up this dying tumor cell material being activated that is dangerous. And then they turn around and they present that to cytotoxic T cells and they activate them. So immunogenic cell death, there's no sense just finding a compound or compounds that kill cancer cells unless you're going to activate the immune system, because there's no way in the world, or I shouldn't say that, it's very improbable that you're going to kill every last cancer cell without killing the patient. Most regimens are used at what's called a maximum tolerated dose. I'm going to make this patient walk the valley of death because I'm going to pump in so much chemotherapy, but I'm going to hope that I'll kill every last cancer cell. And what do we see? We see recurrence. Even the best of breast cancer, prostate cancer regimens, you see recurrent cancer. And it's because the immune system was not activated. It's just like microbial infections, bacterial or viral infections. You never kill every last bacterium with antibiotics. There's always somebody in there that's going to become antibiotic resistant, but you knock down the level of the bacteria to where the immune system gets the upper hand. It's kind of like the Russians versus the Ukrainians, you know, whoever has the most tanks and the most ammunition, etc. But you need that immunogenic cell death to activate the immune system. The real big guns of the immune system are the cytotoxic T cells. And when they're activated, they home and kill very specifically by the tumor peptides that are picked up by the innate immune cells. But that was an excellent question. Is it
Jay, you're
common
muted.
for, I have a chat here. Let's see. Hello, doctor. Is it common for treatments to have side effects? Woo, yeah. Most often is, you know, hair falling out, vomiting, nausea. And if you follow people that are, let's say, children who have acute childhood leukemia, and they're given, you can cure them. You give them 5 chemotherapeutic compounds that are, most of them are DNA poisons.
Dr. Hartmann.
And you pump that in and you'll cure them. But what happens? 10 years later, those children have other cancers.
Dr. Hartmann.
Yes.
That the student was saying that it is common, and if you were able to determine side effects.
Were you able to determine what side effects patients might experience if they took your drug in the future? Well, I'm pretty confident that they would not see. I mean, if you're not affecting normal human peripheral blood mononuclear cells, and the other thing I didn't have time to show you lots of data, we also tested against normal human breast cells, good old healthy normal human breast cells, and no toxicity at the concentration 6 times. Remember, it's 6 times below their effective dose. None of them are DNA poisons, but they're working synergistically. I didn't have time to show you that they work synergistically. That is, you know, some level of killing. It's not just added by the other compound or added by the other compound. They work synergistically. You get a much bigger bang for the buck. So I think that, I can't guarantee that there would not be side effects, but they surely would not be anything approaching doxorubicin or the other anthracyclines and cyclophosphamide, which is a part of most regimens. That's a DNA poison. It cross-links DNA, and you cross-link DNA. DNA is in every cell in the body. It's going to affect the brain. You find that most people who go through chemotherapy, they have memory problems. They have what's even called chemo memory loss. They lose whole sections of their memory following chemotherapy. There's no indication that R3 compounds would have that effect. So Joe's asking, how far along are you with human trials? Wow. Yeah, that's a $1 billion question. So to get to human trials, the average human trial now costs about $1 billion. It's hard to imagine, isn't it? But I just participated in a clinical trial with a Mayo Clinic and two companies doing a immunotherapy trial to try to immunize women for breast and ovarian cancer. Just our part of the trial our part of the trial, we burned through, I don't know, close to $1 million. And that was just a tiny, tiny part of, you have to hire all these doctors to monitor all the patients. You have to have nurses to recruit the patients. You have to have an oversight company watching you to make sure you're not going to do anything crazy. So we would have to, and that's why, you know, in the past, when a scientist would discover something like this, you go, oh, wow, I'll tell the world. I'll go out and, you know, announce this and talk about it. But if you do that, man, you violate any patent rights. It's in the public domain. And now, would there be a company that would even read about it or notice it, or, you know, you have to sell your ideas. So the best approach is, if you discover something, you file a patent. So now that we have a patent pending, I can go to this physician here in town, Dr. Sam Reddy, oncologist, one of the best. And I can sit down and say, Sam, we have a patent pending. And he's part of physicians that belong to a consortium, not only in the United States, but Australia, elsewhere. And they can help us not get all the way to human clinical trials, but raise enough money to do unequivocal mouse trials, and start maybe in another country, South Africa. They don't have all the, you can do a early phase clinical trial over there for like 1/7 the cost of the United States, and they follow all the FDA guidelines, et cetera. So then it's much easier to do clinical trials over here. So we're forming a company, and then we're going to hope that company can raise money through physicians, through so it's good I'm presenting this, because now I realize I have to present it probably in a different way. And I just put this together at the last minute because I was in the lab working on endometriosis today. But so how many billions do we need for
Dr. Hartmann, that's Jay. He's just kidding around.
Jay.
I have like hundreds of compounds that I'm treating on cancer, doc. What do we do with all mine?
Yeah.
Well,
Jay's just playing with you, Dr. Hartmann.
Yeah, well,
But that was awesome. It is awesome that you are finding compounds that can kill off cancer and do leave other normal cells intact. And we will, if you guys do have questions, feel free to enter them in the chat. We do need to move on to our next professor.
Okay.
So I know I don't want to speak for him, but I know Dr. Lee does stay up late regularly. So I don't know if Dr. Lee minds being last, but Dr. Toll, maybe you would like to go now?
I stay up late too, but sure, I'll go next.
All right.
My talk's going to be very different.
So Dr. Hartmann, if you can.
I stopped
flip
sharing.
the-
Yep. There we go. Wow. Big audience here. Thank you all for coming and listening. And
All right.
now let's
Let me get here. I'll share my screen. Share screen. Share screen. There we go. I think.
Yep, something's happening.
Oh, something's happening. Let's go here. There we go. So
Excellent.
first of all, I'll clarify a few things. I'm not a chemist. I'm not trying to derive new naloxones.
Huh.
And so everything Michael said was wrong. But other than that, thank you for the introduction. And I go by Larry, so you have to call me Larry. When Michael asked me to talk, I didn't really know what I was going to talk about. So I said, you want me to talk about, he said, the new research you're doing, well, there's a lot. So I said, do you want me to talk about what I do for a living? He said, sure. So that's what this is about today. So I'm a pharmacologist. I study opiates and other drugs of abuse. Opiates we study with respect to both pain and drug abuse. As you know, opiates are the best analgesics, the best painkillers. And so my lab does in vitro work and work in animals and imaging studies. And we try to understand mechanisms by which opiates and other drugs of abuse work, and neuronal pathways that mediate their effects. And we try to design new drugs for the treatment of drug abuse and pain, and new analgesics. And so that's what I'm going to briefly talk about today. And there's a lot there, and we'll move through it pretty quickly. The first thing I want to talk about is that what we're looking at, and these are morphine or opiates and cocaine. And a lot of people have asked me if they're the same, right? You know, they come from the same place. If they do the same thing, the answer is no, completely no. Opiates, morphine, and opium come from these things here. You know what? I'm going to think I can do this. I can do a laser pointer. I learned that today. I can teach you if you want to learn how to do that.
Yeah.
So opium comes from an opium poppy. These are grown all over the world, lots in Afghanistan, Mexico, other places. And you can see this white stuff that comes out of the poppy. This is opium. And you make this is the bulb of the poppy, and you make a little cut, and it's so full it just drips right out. And morphine is a natural product. It comes from this sap that comes out of these poppies. There's other opiates in there as well. Codeine is also found in these, but the major constituent, in fact, is morphine. And so this is purified. It's never synthesized. It's literally all the morphine in the world is made from these poppies. Cocaine comes from the leaves of a coca plant. So these are found in different parts of the world, and you grind up the leaves and you can extract the cocaine from these plants. So they're different molecules. They come from different sources. So as I said, I'm a pharmacologist. I work with chemists a lot and always have. And chemists see these structures. It means very little to me, though I've learned a bunch about it over the years. But this is the structure of morphine here. And you can see that there's been an incredible amount of medicinal chemistry that's done on this class of compounds. And that's because it's a super important class of compounds, right? Opiates are the best analgesics in the world. They kill pain better than anything. And they've been used for thousands of years for this exact purpose, literally thousands of years for this exact purpose. And other things as well, because there are side effects induced by opiates like constipation. And in fact, the Egyptians used morphine, opium for diarrhea. So it's used for lots of things. Huh. And there's been a lot of work on morphine, and there's been a huge amount of chemistry that's done. And you can see that the compounds that are made, some of which you've heard of, like heroin, is a morphine analog. This was the first one made. I have my slides that show this was actually made by the Bayer company, Bayer. They make aspirin. They made heroin in the start of the the end of the 19th century, and actually literally sold it as an analgesic. But you can see that these guys are made from morphine specifically, and then there are other compounds that are fully synthetic, some of which you've heard of, some that you haven't. Fentanyl you all have heard of these days because it's super easy to make. It acts like morphine, only it's 100 times more potent. And the good news is it's way more potent, so you don't use much of it. And the other good news is it's easy to make, so you can make it. The bad news is it's easy to make, so it can be made in little shops in Mexico and brought into the United States, and it's really bad for you. Anyway, these guys all act by binding to opioid receptors, and that's what I study. I study receptors. This opioid receptor is called a GPCR. That stands for a G protein coupled receptor. This is the largest family of proteins in the body, GPCRs. And half the drugs used to be, now at least a third of the drugs in the world that you ever take are active at these things. They're either agonists, they turn on these receptors, activate the receptors, or they're antagonists and they block them. And this is, so this is a normal, the opioid receptors are normal, run-of-the-mill GPCRs. These are all also called 7-TM receptors. As you can see here, this is a sort of called a snake figure. And you can see that it passes, this is a membrane in a cell. and this passes through the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. So this is a 7 transmembrane receptor. And this just shows you there are multiple different subtypes of opioid receptors, and they have very high homology. They're all in one family. And I'll talk about this in a second. This is actually the crystal structure of a receptor. Crystal structures have been fairly recently identified for GPCRs in general. And so this is what the mu opioid receptor looks like. And inside here, where this green stuff is, that's where the ligand binds. That's where morphine binds to the receptor. And you can see, in fact, it's not here on the outside of the cell where you would think that it might likely bind because there's room. In fact, there's a cavity inside these things, and this is where the ligands bind, inside the 7 transmembrane region. And in fact, this is the place where there's highest homology among GPCRs. And that's because there's a tight binding between the receptor and the ligand. And that's how it works. This is how they bind. So these are called GPCRs because G protein coupled receptors, because they couple with G proteins to mediate a signal. So once the ligand binds to the outside of this receptor, to the inside of the receptor, it increases the interaction of the receptor with this intracellular protein called the G protein. And I'm not going to go into the mechanism by this what happens, but just to say that it affects things. So in fact, once an agonist binds to the receptor, it activates the G protein, which then the first things that were discovered is they interact with a protein called adenylate cyclase, which is used to manufacture cyclic AMP, which is the super duper intracellular signal for things to happen inside cells. And so that's what happens. One of the things that happen when the GPCR binds an agonist, it causes this coupling to the G protein, and then either a stimulation or inhibition of cyclic AMP. And so that's what it means when it's a G protein coupled receptor. Okay. Opiate receptors, it turns out there's not an opiate receptor like we thought when I was starting to work in this field. And I'm old, almost as old as Jim Hartmann. And it turns out there's 4 receptors in this field. The important one for us today is the mu receptor. It turns out that the mu receptor is the one that mediates the majority of the analgesic actions of opioids. And so here I wrote the actions of the different receptors, but the most important for analgesia is this mu receptor. And in fact, every drug that's an analgesic that you've ever taken, that anybody's ever heard of, these are mu agonists. Okay, they activate this. Now these receptors are present in the membrane, and they aren't there originally. Your body didn't develop and design these to be activated by something that comes from a opium poppy, obviously. So that means there must be an endogenous ligand that binds to those opiate receptors. And these ligands are called generically endorphins. You've heard of endorphins, right? Endorphin stands for endogenous morphine. That's where the word comes from. And in fact, endorphin is a generic term that means endogenous peptide that binds to opioid receptors. And in fact, there are various families of these ligands that bind to the receptors. There's various families of these receptors. So this family of receptors are related. This family of ligands are related. And they are also related to other neuropeptides and other GPCRs, but not as closely as they are to each other. That's why this is a family. So it turns out there's 4 receptors in the opiate receptor family. The one that, as I said, is most important for analgesia is this mu receptor. The one that's most important to me personally, in fact, is this 4th one called the ORL1 receptor, which stands for opiate receptor-like receptor number 1. And that's because when I was in sabbatical in 1994-95 in France, I was involved in the discovery of the endogenous ligand for this thing, which we called nociception, which was also called orphanin FQ. Anyway, so this is an endorphin in a sense as well, because it binds to one of these receptors. It's different because it doesn't bind to the other three. And these ligands, these endogenous ligands actually bind to all three of these, but don't bind very well to this fourth receptor. And nociception, orphan FQ, binds to this fourth receptor better than to the others. Nevertheless, they're in the same family structurally, and they're very important. And this fourth one is also involved in analgesia and in drug abuse as well. So this is the system in which I'm working. Okay, as I said, I'm looking for compounds that are analgesic, that don't have the problems associated with normal opiates. The biggest problem, of course, that you all know is reward and abuse liability. And so Michael was correct, is I do still work with other chemists, and we are looking for drugs that would be the gold standard for an analgesic, which would be something that would be a powerful analgesic like an opiate, but non-addicting. And so this is work that I do, and I'll talk about it in a second. I mentioned that opioid peptides, and as I said, these are a family. The important thing to know about these is they have a similar structure, and enkephalin is something that I had mentioned previously. This was the first one discovered to be an endogenous opioid, and it's a pentapeptide, 5 amino acids that goes tyrosine, glycine, phenylalanine, and either leucine or methionine. And in fact, all of the opioid peptides have this same structure, the same sequence. The fourth one, which that I worked on, which nociception, orphan FQ, in fact, has a phenylalanine here instead of a tyrosine, which in fact is why it doesn't bind as well to the other receptors. That's a story I'm not going to talk about today. And all this other stuff here, ignore it. I'm not going to talk about that. All right. So this is the system in which I'm working, and I'm going to talk briefly about what we do and how we do it. So this will be broken in two parts. I hope I have time to do this. One is drug discovery, as Michael talked about, and the other is pain mechanisms and circuitry. And in my lab, we do both of these. All right, to determine if something is a good ligand for something, there's experiments that we have to do to show that it works, okay? And in the olden days, to see if something was a good analgesic, in the olden days, they would give it to a person and say, do you feel better? Literally, that's how heroin came about, okay? And there was a paper that I have read that I have in a different slide that talks about drug discovery in, I think it was like 1955, when they were doing the very first studies on mice to look for analgesic activity. This has moved forward since those days. And in fact, the first experiments that we do now are in vitro experiments because they're much easier and faster, just to see if the thing you're working with binds to the receptor that you're working on, or activates the receptor, or inhibits the receptor. And so we do something called receptor binding. This is a very simple assay that you do in a test tubes. We do it now in a 96-well plate format and use this cell harvester thing. And we use radioactive ligand that binds to the opioid receptor, and we see if the affinity at which it binds to the receptor. And so you can screen lots of compounds this way if you're using a 96-well format. You do this kind of an experiment. You can do an experiment where you increase the amount of the radioactivity, and you should see a curve that looks like this. So by definition, it should be saturable, right? It should go to high concentrations and not bind anymore. And by definition, from this, you can derive the affinity of the ligand for the receptor. The dissociation constant, by definition, is the concentration at which you have half maximal binding. So you can get that from this curve. And then you can also derive the B max. This is the maximal number of binding sites in the preparation that you're using. So these are important parameters that we derive. Then you can do a similar, slightly different experiment if you want to test lots of compounds. Unfortunately, you can't radio label every compound you want to test. So you do a different experiment where you take the radio labeled compound and you use an unlabeled compound and you displace it with various concentrations of that drug. And from this, you can derive an IC50. which is a function of the affinity of the non-labeled drug for the receptor. And so by this mechanism, we can test lots of compounds. And you can throw out ones that don't bind, and you can take ones that do bind. We're looking for things that bind with nanomolar affinity. Okay, this is not like methotrexate and those things where you're using micromolar compounds. These we're looking for things with high affinity, nanomolar affinity that bind to the receptor. And from this, we can screen quickly, and we can find lots of compounds that bind or don't bind and move forward. There's also in vitro activity and in vitro assays for efficacy. So we can tell if something is an agonist or an antagonist. So if it activates the receptor by opiates, in fact, inhibits cyclic AMP, so we can measure the inhibition of cyclic AMP in a cell system and determine if something is an agonist or not. Okay, these are all important studies. So from then, if you're going to look for a compound that's actually an analgesic, you can't tell from these in vitro assays what it does, obviously. You got to stick it in an animal. And we do these studies as well. And we use normal assays that people developed, some of them years ago, some of them more recently. And so we look for pain in a mouse or a rat. And the most common assay, in fact, is called tail flick. It's just what you might imagine. You make the rat or mouse flick its tail. The way you do this is by heating it. So some people use warm water. You literally dip the animal in warm water. And we use light, heat from a light source here that we can shine on the tail of a mouse or a rat. And you literally measure the latency in which they flick their tail. And so depending upon the amount of heat that you're applying, an animal will flick their tail in a few seconds, 2, 3, 4 seconds. You can do the same on a person, by the way. With all our students, we do the finger flick. And you put your finger on the light and you see how long it takes them to move their finger. And you can see this is not an invasive test. It's easy, doesn't hurt anything. When it gets too warm, they move their tail. Unless, of course, you give them morphine, because then they won't move their tail, because morphine works. And if you give a dose of 10 milligrams per kilogram or 15 milligrams per kilogram, they'll leave their tail there until it burns. So we have a cutoff of 15 seconds so we don't destroy the tails of these animals. Another test that's normal is a hot plate. This is literally a hot plate. Okay, you set a temperature of 52 degrees or 55 degrees. You put them on here and you wait until they lick their paw and you measure the latency. And so this is another experiment that we do that shows the efficacy of an analgesic. And again, you don't wait until they burn their paw. If when they lick their paw, you take them out. If you give them morphine or another analgesic, obviously they'll leave it there longer, and this is what we measure. We also do chronic pain studies. Chronic pain these days is just maybe more important than acute pain. And there are the various models that we use. There's one important one called chronic constriction injury, where you tie up the sciatic nerve. We do something called a spinal nerve ligation, where you cut off the nerve as it gets up to the spinal cord. And what this does is it causes pain in the paw if it happens to be this nerve. So the sciatic nerve, you've heard of sciatica, your leg hurts, right? This is tie off the sciatic nerve, or the spinal nerve that approaches the spinal cord, and it causes pain in the paw. And you can measure this in various ways. We use this method here. These are von Frey filaments, it's called. And you can poke the animal in the paw. And if it has this peripheral neuropathy caused by the surgical thing that we do, it's more sensitive, and you can poke with a finer paw, and it'll make them move their foot rather than a thicker paw that you need to do an animal that didn't have the surgery. So this is a model that we use. You can also do heat, and they'll move their paw faster because they have heat allodynia, or this is called allodynia, which means it's a painful response to a normally non-painful stimulus. So there's different methods that we use to look at this chronic pain in animals to measure this allodynic response. With an opiate, you have a side effect, of course, that you have abuse liability. And so we have to test this we have various models to test this. This is a really interesting test called a CPP, or condition place preference. And the way this works is you have a box like shown here, you have one side that's different than the other side. Okay, so this has bars and this has slats. This is one color, this is another color. And the way you do this experiment is you take a rewarding drug like morphine, or one that we're trying to develop, and you put it in one side and you let it sit there for 15 minutes and you run around. And then the next day you give it vehicle and you put it in the other side and you give it 15 minutes, you let it run around. And you repeat this process for a few days. And then on the last day, there's a little door here and you take out the door, you put them inside and you say, go. And then you watch them for 15 minutes and you time the amount on either side. And as you can imagine, if the drug is rewarding, they will spend more time on the drug paired side. Okay, so this is a test we look to see if something is likely to be rewarding. And so this is a standard procedure that people do to determine if a new drug they're testing is going to have abuse liability. Another test that we look is at drug taking behavior directly. And this is called self-administration. You can imagine how this works. You give a drug that you're administering through a catheter in the jugular vein, and you put it in a box, and they have a lever to press. And if they like the drug, they will press the lever. And if they don't like the drug, they won't press as much. And so it's the same type of procedure you put in there, and you can test a drug to see if it's likely to be self-administered. By the way, this can also be used to determine if a drug can be used as a drug abuse medication. And we do these exact studies as well. And so if you give a drug that blocks the actions of a rewarding drug, what you'll see, in fact, is a rewarding drug like morphine, boom, 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 they take a drug every day, they'll press the lever 25 times. But if you give the other drug on top of it, they'll stop, okay? And so this is an indication that the drug may be used as a drug abuse medication. You know what? I think that's all I'm going to talk about today. I've taken my 20 minutes, my 25 minutes. So I'm not going to go on to the pain mechanisms and circuitry. I'll suffice it to say, we look at the different cells that are in specific cells that are involved in this pain experience. This is the nerves that I was just talking about. This is something called a dorsal root ganglia, which is the cell body for this nerve that goes literally from your foot to your spinal cord. And we can look at the different layers of the spinal cord, and we can look at what cells are literally being activated. You can see here, well, this is, I can show it better here. So this is a picture of the spinal cord, and looking at the different receptors. This, in fact, is the receptor that I was talking about, the NOP receptor, the one that I'm most interested in. This is the dorsal root ganglia, and you can see the individual cells here. These are cells are NOP receptor. You can see there's a lot of them in the spinal cord, in the dorsal root ganglia, and in these neurons that go to the spinal cord. And we have a method that I'm not going to talk about, except to say we can actually directly, using this mouse models, we can actually directly look at cells that are in fact activated by the pain. And this is the spinal nerve ligation model that I was talking about. And if you poke them in the paw, in the sham animals, the cells that are seen here in red are actually cells in the spinal cord that are being lit up. And you can see in a normal animal, when you poke with a very thin fiber, you only get a few cells lit up. But if you poke with a spinal nerve ligated animal, it hurts. And you get all sorts of cells that are lit up. And the cells in different lamina of the spinal cord are important for different kinds of things. And what we're interested in doing is really understanding these specific pain-mediated, pain-causing cells more carefully. And we're looking at the important brain regions for pain, from the spinal cord to what's called the RVM and the PAG, parietal-actogallical gray. And you can literally see cells in these regions that are important for pain, that are turned on by pain. And we're studying these cells directly to not only understand the circuitry involved, but also if you study these specific cells, you can see what other types of proteins or receptors are present in these cells that you may be able to use in the future as drug abuse medications. And there you go, that's all I have to say. These are the people who are doing these. I got a nice big lab that does a whole lot of things. And anyway, if you have questions, because I know I went really fast, My door is open every day. I'm in the School of Medicine. Thanks. Goodbye.
Thank you so much for your presentation, Dr. Toll.
Very nice.
Any questions?
There is actually one in the chat. They're asking, would the next step from the mouse trials be primates? Because I know that certain analgesics affect rats differently than that of humans.
Well, that's a really good question. And the answer is, in the olden days, no. These days, I think it is more common, though not always do people do this. But yes, the things that we've used have all gone into monkeys as well, into non-human primates. And there are differences. In fact, compounds that bind to the receptor that I'm interested in, NOP, it turns out don't work very well in rodents, but work very well in primates. In general, the receptors are quite similar. And what's more different, in fact, between rodents and primates in general is the pharmacokinetics of the compounds. And so things don't work very well in primates, but might work better in humans and primates is because of the PK, the pharmacokinetics. So, but yes, a lot of drugs are tested in non-human primates as well as in rodents. It's just obviously much easier and faster in rodents, cheaper. Anything else? I may or may not know an answer.
Well, I have a kind of a, I don't know if it's a good question or not, but it's a question. As an immunologist, when we think of pain, we think of inflammation, immune cells releasing inflammatory cytokines. So where's the connection between your studies and would you entertain maybe looking at the release of inflammatory cytokines?
So neuroimmunology is an important field. There's a guy named Ning Quan, who is in Jupiter, who does this for a living.
Oh, okay.
We now have a grant together, in fact, where we are looking at that exact question. So the answer is, for opiates to work, the immune system does not have to be involved.
Mm-hmm.
So opiates will block any pain stimulus, whether it's an inflammatory pain or neuropathic pain. Okay?
Oh, okay.
We actually do inflammatory pain models. We use CFA. Do you know CFA?
Yeah, complete foreign judgment. Yeah.
It causes a massive inflammation, right?
Yep.
And it really hurts. And if you give opioids, you block the pain. It doesn't do anything to the cytokines, right? It doesn't do anything to the inflammation, but it blocks the pain anyway.
Huh.
It's because it blocks the pain signals from the paw or wherever it is to the spinal. So opiates work anyway. There's a long history of looking at the interrelation, moderately long, between cytokines and opiates. And in fact, the story now is that opiates increase IL-1 beta, which causes pain.
Yeah.
And so if you block IL-1 beta, it actually potentiates the actions of opioids. So opioids are working against themselves by increasing cytokines. So there is work on this, but the bottom line is opiates don't need it to be analgesic.
Very good. Thank you.
Okay. Sorry for talking so fast.
How did you spell that guy's name in Jupiter? Nin Quan?
Ning, N-I-N-G. Quan, Q-U-A-N.
C-U-A-N.
Yeah, he's in our department.
Oh, okay.
Dr. Toll, I have a quick question. In your tail flick assay, what is the difference in temperature that's required for you to see the tail flick when you give a rat analgesic versus when no analgesic was given?
Well, we don't look at a difference in temperature. We look at a difference in latency to when they flick.
Gotcha. Okay.
So the temperature is the same. The time is different.
Gotcha. All right. Thank you. And you also look at you said you use a hot plate, and do you mind going back to that slide, because it sounded really interesting. I just got a little bit confused because you said you used a hot plate and you used a heating lamp, I believe is what you said.
So we use, yes, we use radiant heat for the tail flick.
Gotcha. Okay.
So if you look
Sorry
here,
for making you go back.
no problem. Go. So this shines a light, and so it's radiant heat. It's warm. If you put your finger there, you're going to want to move it after a few seconds. And so we shine it on the tail. A lot of people use warm water. It's probably a little easier on the tail, but it's warm. You make it 52, 53 degrees. If you put your finger in it, you're not going to want to leave it there. And so that you just literally measure the time at which they move the tail. The trouble with the warm water tail flick is you have to stand there with a stopwatch, and you got to literally time it with by yourself. If we're using this method, this is hooked up to a timer, and so as soon as the mouse or rat moves their tail off the light, the timer stops.
Gotcha.
Okay. So to me, it's much more accurate.
Okay, thank you.
Again, with a hot plate, you have a stopwatch, and you got to measure the time.
Right, and then remove. Okay, thank you.
Sure. Do you mind if you share my, of course not, share my email.
All right. So guys, like I did Dr. Hartmann, I will share Dr. Toll's email in the chat if you're interested. And if there's not any more questions at this moment, feel free to add them in the chat, but we will be moving to Dr. Lee's presentation.
I enjoy let me see, open okay, so I enjoyed very much the two research talks. My presentation probably have a lot. So let me share slides here. So I think the first part of my research, my slides, I think are good for the seniors and juniors undergraduate students. So that's the level I'm going to talk about. For the second part, I have a few slides about my own research. Yeah, so just next. So I guess most people know about most general population people know about precision medicine for a few years ago when the government started a precision medicine initiative, which is to sequence lots of people genome sequencing. If you're interested, you can see that there is a video here. So what is precision medicine? According to NIH's definition, it's a groundbreaking approach to disease prevention and personalized treatment based on that person's individual difference in genes, lifestyle, and the environment. So this idea is not new. It's many years ago, since from the blood transfusion, that is the original idea for precision medicine. And I guess many of you probably also know about 23andMe. With $99, they can tell you the risk for example, for Alzheimer's disease risk, you have 7% risk for that. So they use the SMP, single nucleotide polymorphism markers, which found from the literature, report certain markers associated with a disease. They use these markers to predict disease risk. And so, for example, for the alcohol dependence, I think they used some markers from papers from my lab. And so the goals of precision medicine is to use the right dose of the right drug for the right person at right time. And so today I'm going to give you 3 examples about precision medicine. There are a few others, but those three are because they are related to genomics, which I'm doing. First is predictive medicine. So you all know that for the Mendelian disease, you have the mutation, you have the disease, right? That's simple. So for the most of the newborn screening, that is use those Mendelian markers to predict. But that is new. But today I'm going to introduce one example about the complex disease. The complex disease, there are many genetic markers, genes are involved. How to do that? So this is one example, a genome-wide association study. Sorry about that. So this is a disease for eye disease. Typically, that's what we see. Patients with this disease, this is what they will see. So a group of people, if they did a GWAS, they found here 5 SNPs associated with this disease. And so there are 5, but for the patients, not everybody have the same profile, genetic profile. Some people only have 1 mutation, some have 2 mutations, some have all the mutations. And because we are diploid, 2 copies, some only have 1 copies, the other have 2 copies or 0 copies. So those are the different scenarios, how the combinations of the mutation allele you have. So the higher number of mutation alleles you have, the higher risk you have. That's the idea. So those 5 SNPs together explain half of the disease heritability, which is very high for most complex disease. Probably there could be thousands of genes are involved. So for this specific disease, the GWAS study, genome-wide association studies, was very successful. Only 5 SNPs, they explain half the heritability, which is very good. So using the 5 SNPs, we can predict the disease risk. This is in the general population, what's the risk you have the eye disease as you get older. And now, if for people who have the certain combination of the mutation allele, that is the risk. You can see the very different. So this examples, we use the genetic variants we found from the genetic study. We do the disease risk prediction. Actually, there are already quite a few companies, they apply the patents to do the genetic testing and disease risk prediction. This is another example. So we know the BRCA1, BRCA2, the 2 tumor suppressor genes. For the general population, that's the risk for breast cancer. For BRCA1 mutation, that's the risk. For the BRCA2, that's a big risk. This is for the ovarian cancer. I guess probably you all know this brave woman. She has a mutation for the BRCA1 mutation. So based on the genetic testing, she removed her mammary and the ovarian tissues, both of them, and so that's one of the examples. Now I'm going to talk about the pharmacogenetics. This is a study to study how the genetic variants affect the drug metabolism and transporting and their safety issue. So this here is one example. This drug was designed to treat the leukemia and other autoimmune disease. But some patients with a certain genetic mutation might have the high side effects, including toxicity. And so this is one gene, this TBMT gene. So this white tab, so the enzyme, the protein encoded by this gene is normal function. If some people, they might have mutations here and there, so those mutant tab of the gene, so the enzyme function is not good, dramatically decreased. So that's a problem. The drugs cannot be cleaned out from the blood. So for the white tab, the toxicity is very low, but for the 1 carbon mutation or 2 carbon mutations, the toxicity is very high. So traditionally, when we treat a disease, the clinicians just give the drug the same dose according to that label, the same dose. But for those group, this subgroup of patients, if they have the mutations in this gene, so they have very high toxicity. This is conventionally what we did. But now this is what we should do. So what we should do is before the clinician give the drugs, the dose, so the clinicians first should do other genetic testing of the mutations in this gene. If one person have the mutation here, that's the dose the clinician should give. You can see very different, so that then keep the same level of the drug in the bloodstream. So that to prevent the strong extremes toxicity. So this is a different drug. This drug was designed to control the cholesterol, but too much of the drugs may induce the myopathy. So we also already know mutation in this gene will induce very different side effects. This is a wild type. This is 1 copy mutation, 2 copy mutation. You can see that's what the side effect is very big, very strong. So there are many other genes that we should clinician should do a genetic testing before the drug. And so I follow the FDA actually has those information online. So I check that almost every year we issue. You can see the number of drugs who have the labeled such genetic requirement, genetic testing requirement. Is you can see the numbers are increasing year by year. So this is about how we use the existing genetic variants which we found from genetic study, how to use this for the genetic prediction, disease risk prediction I talked about now is pharmacogenetics testing. So now the third part I'm going to talk about called targeted cancer therapy. So conventionally for the cancer patients, typically in the past many years, clinicians treat patients according to where is the cancer cell from. This breast cancer, they have a protocol. For the lung cancer, they have different protocol. Now what we are doing right now should it be is, so for all the patients, we need to do a genetic testing, molecular diagnosis of genetic testing, and then based on the driver mutations, then we choose the treatment called targeted cancer therapy. If this group patients, no matter it's breast cancer or lung cancer, as long as they have the mutation in this gene, then you should use the therapy which was designed targeted to this gene versus the gene B or gene 3. If there is no information, then we have to use the conventional treatment. So one example about lung cancer, the lung cancer mutations, a portion of the lung cancer patients, they were caused by the mutation in this gene. So there is already drugs designed to inhibit this pathway. So now this is what we should do. The clinician first to order genetic testing to see if the patient has a mutation in the EGFR or not. That's a very simple PCR single sequence testing. And then based on the result, if you have the mutations, then you should use a different one. This is a clinical trial. This is called a survival curve. This is conventional treatment. This is a cancer targeted therapy. So you can see this is a 1 year point. At a 1 year point, you can see the patients who received the conventional chemotherapy, almost all of them died. However, for the new treatment, about 40% of them are still alive. So they demonstrate this gene targeted therapy is good. And this is a very similar story for the breast cancer, for example, HER2, that's the 11% of people, they have HER2 mutations. Similarly, there is a commercial drugs that is antibody against the HER2. Now, this is what we should do. The clinician first should do other genetic testing to see this. Normally, we should begin the HER2 gene. We should have 2 copies normally. But this is for the patient, breast cancer patient. If they have HER2 amplification, that's what you see of the proteins. You can see here the brown is on the surface of the cell. Lots of HER2 proteins. The brown, that's proteins. The thousands of copies. And also on the genome level, you can also use the fish. You can also see the many copies of this. So based on this result, so that's what the are the different treatment. So this is similar to the survival curve. You can see roughly using the treatment design for HER2 can basically save 10% more patients of lives. So this is what we should, I think, in the coming many years, that will be genetic testing, genetic research will push the genomic medicine eventually to the personalized treatment. And eventually you go to see a doctor, you get your drug, you will probably swipe the chip, ID chip, to see what's your genetic profile before you get a treatment. So this might be associated with a disease.
So it could differ from patient to patient.
Oh yeah, they're very different. We find
with the same disease, but a different curve.
Oh yeah, could be. That's we don't have a solid answer right now, but yeah. So the people, I think, for example, for the ME/CF, it's a syndrome. It's not one disease. Actually, in the past few years, I think we were trying a few years ago. We also did the genomic sequencing. We're trying to do the linkage mapping. We see if there is any mutations that cause the disease in the family. But no, this is we see this is now. I understand this is not a Mendelian disease. This is a complex disease, and it's a syndrome. Yeah. So.
Wow. Whew. I didn't realize how common it is. My God, it's terrible.
Yeah. That's before COVID is 1%, and now is because the COVID long hauler. So far, we don't know if the two COVID long hauler and if they're same disease or 2 different diseases. But at least on the symptoms, 95% of symptoms are overlapped.
Unbelievable! Wow. Fantastic talk. Very fascinating.
Dr. Lee, I have a question. Do you think that, given some of the symptomology, do you think that CFS and long haul COVID, so to speak, if they can be used as models for each other or one or the other?
Yeah, this is a very great point. Yeah, that's what many people are thinking. Because without COVID, actually, we don't know what triggered the ME/CFS. So people in the past are trying to do to capture the virus infection. Even including my lab, we are screening for viruses in the blood. But we could not find we could not find one virus that commonly exists in all patients. No, we could not find that, because I think that's easy to explain, because the initial virus that triggered the disease might have already been cleared by the immune system many years ago, because most of the patients have the disease already for 20 years, 30 years. The initial virus is gone. We cannot capture them right now. So now the COVID, now we know the COVID triggered this symptom. We know which virus. I think they give us a very good model. This is a disaster, but now they give us, for research, they give us a very good model. We can track down what could be the cause and the consequences. I think this is that's what my lab also actually we're developing also similar research as what we did in the CFS. We are doing a similar thing in the COVID long haulers. Indeed, we are collaborating with the people at the FAU. We collect the COVID long hauler patient samples. We sequence them and yeah, that's what we're doing. Um,
I have a wild suggestion. You two guys, Larry, I don't know if Larry's still here, but it'd be interesting to see in long COVID or ME, if you take the antibodies from those patients and you screen them in Larry's 96 well brain tissue to see if you can pick up any antibodies that have been elicited by the COVID or by a HERV that are now binding to brain tissue. And that's very similar to what Michael Hamper and I are working on. But it seems like you two guys could get together and maybe uncover some, because when you get the immune response, you're going to have a universe of antibodies. And so to try to sequence or PCR, I mean, very, very difficult, but you could screen and try to find if there are antibodies present, and then what are they binding to? And you probably find again differences from patient to patient in terms of the antibody. Just a wild suggestion or a hypothesis.
Why, this is a very good suggestion. Yes, I thought about this for long, about to test the proteins, antibody proteins, but my lab is limited because I don't have bench. So I need a.
Oh, that's right.
My lab is.
But you have access to patient serum. Do you have access to patient serum that you send out to?
Yeah, we do have patient samples. We have whole blood.
Oh, yeah.
But I do need collaborators who can test it, who can do some test. For some easy test, I typically order through the core facility. But for advanced research, I do need to work with the collaborators. So that's why we now work with I have a few collaborators. Now I'm excited about FU here. We have labs from different disciplines. We can work together. Larry is not here, but I should talk with him.
We ought to all get together for lunch. And I do a lot of immune testing, immune assays, flow cytometry, that kind of stuff for individual cells. We can even screen the antibodies for reacting to neuronal cells. Michael Camper and I, we have a neuronal cell culture in the laboratory, so we can even look at the surface, or we can look inside the cell using flow cytometry to see if any of those antibodies are binding. But yeah, we ought to get together with Michael and Larry and then have lunch. Now we don't have to wear masks anymore. Hopefully we could actually talk over lunch. So we'll try to get together when we can all find some time.
Yeah.
When are you guys primarily available if students ever want to reach out to you or come into you and see
I'm
your office?
Monday, Wednesday, or Friday. Good for me. I teach Tuesday, Thursday.
I just the best way for me, just send them an email. We can.
Yeah.
Give you the advice.
Yeah. What's that thing you send around? Google Scheduler, and you just indicate your times available. I forget what it's called.
Yeah, actually, I'm very happy to know here you guys have a student organization. So I admire you guys have like peers co-mentoring, the co-study. I think this is a very great thing. Yeah.
Does anybody else have any other questions for these two?
We should take a poll and see how many people actually believe that Fauci knows what he's talking about. I certainly do as a scientist, but there's been so many. He's afraid for his life, his family, his children. because there's so many people are attacking him because he's trying to save lives. It's incredible, isn't it? That a scientist could come under that type of attack. I don't know if you heard that he's come out publicly saying that he fears for his life, his wife, and his children because he gets death threats.
Wait,
Absolutely crazy.
I mean,
It's like, it sounds like you getting attacked to get into your lab.
Sounds like what?
It sounds like you previously students trying to get into your lab.
Oh, that they've sent a chat or something, or students that want to get into Dr. Lee's lab?
wait, I'm confused. What happened?
Oh, I was just rambling on about, because Dr. Lee mentioned Dr. Fauci said that the long haulers, that it's suggestive of ME.
Hmm.
And so I was just kind of curious if we have any anti-Faucis,
Oh,
non-scientists in the audience, but I would certainly hope not.
yeah.
But who knows? There might be some Trumpers.
Actually, Dr. Lee, quick question. Have you seen any difference in those with, or think that there is any difference in those that have gotten vaccinated against COVID and the comparison with those in ME?
Good question.
Yeah, so COVID, yeah, we did, we are still doing, we are still testing, but what we finished one is about epigenetics. We did see a difference between the COVID long and ME/CFS. We did see the difference. We're still working on, we're still analyzing the data. We don't have any conclusion right now, but they are definitely, so this could, we did see the epigenetics difference. There could be many explanations, because it could be one disease, but just because of different stages. COVID long is in early stage. The ME/CFS has already 20 years. They're two very different stages. So we are still working on that. We don't. I think too early to talk about it too much right now. We're working on that. But I think it's a consensus that in the field, people expect there will be a pandemic of ME/CFS occurring from the long haulers.
Wow. Wow.
Because there is a percentage, a portion of the ME/CFS long haulers, they already match the current criteria. That's half percentage. And we know in the ME/CFS is once people are diagnosed with ME/CFS, there is no cure at this moment. Yes, it's very sad news. It's a very sad thing.
Yeah. Wow.
I've been talking with many patients in the past, and before COVID, I talked with a lot of patients. They are also, you can from their eyes, you can see they're looking for a cure. They're looking for something, but they were misunderstood. I mean, they said that the clinicians do not. Many clinicians thought this is, you guys are lazy. You can just do more exercises. They thought this is a psychological problem.
One thing that might be interesting would be to see if, like multiple sclerosis, there's a very strong north-south gradient. And so if you're born and raised in Florida, your risk of multiple sclerosis is very, very low. If you're born and raised in Michigan or Canada or Upper Peninsula, your risk for multiple sclerosis is very high. And now we understand it's because if you live in Florida, you get sunshine, you capture the energy in the sun and you make an anti-inflammatory steroid, but we call it a vitamin D. So I would hypothesized that many of these people that have ME, you could screen we could screen their blood to see if they have low vitamin D levels, because it's almost a given. 70% of the American public has either deficient, I mean zero, or they're insufficient in a natural anti-inflammatory steroid. And it predisposes to, I published a paper on asthma. I mean, name the disease, because most diseases have a highly inflammatory component. My hypothesis is that the majority of, well, it's almost a forgiven conclusion. If you were to assay their blood, you would find that the majority of them probably are totally deficient. I mean, zero. But most clinicians say, oh, vitamins, you get all that in your diet. No, don't bother me with that stuff. And it's too bad it was mislabeled a vitamin. It's the safest, most powerful. In our study in asthma, we showed that vitamin D3, even the storage form that you make or that you take a pill, is nearly effective as an anti-inflammatory as dexamethasone. But do you find doctors giving vitamin D? And there are even very powerful, these people should be treated with a non-calcemic analog of vitamin D. Very safe, very powerful, minimal side effects, no calcification. Because if it's an inflammatory, and I think the same thing is going to be shown for the long haulers. Many of them are going to be deficient or insufficient. And we know that whole story from multiple sclerosis, the north-south gradient. I mean, it's very well established in the field of multiple sclerosis. So
Someone asked,
definitely got to get together for lunch, and we could do some vitamin D levels in the patients and see what their level is. I mean, it doesn't, if they're deficient, it doesn't absolutely prove it, but what you might, if you catch somebody early stage, and you give them a therapeutic dose, not just, you know, a minimal amount in a multivitamin, but a therapeutic dose of this natural, safe anti-inflammatory, you would turn their life around. I would bet on it. I'll bet you $100, maybe more. I mean, but
that is definitely a grand statement that I think a lot of people are going to think about.
Yep.
Dr. Lee, someone asked a little bit of an epidemiology question. Does that 1% of population that has ME, does that include people that are undiagnosed or maybe not show symptoms?
Very good question. I think this 1% is more like estimated, because it should include the people who are not diagnosed, because we don't know the actual real number. Because so far, what we know is most patients, they are not diagnosed yet. As I know, there are only a handful of clinicians in the United States can actually actively diagnose the patients. There are a handful. Huh. Those people, I know most of them, just not too many. You can imagine the most of the patients are not yet. So far, if someone wants to diagnose this patient, they might take a flight to different state to diagnose. Many states don't have even one clinician who can diagnose, who have experience to diagnose.
Is there a diagnostic biomarker? So he goes on symptoms. The clinician goes on
symptoms. Yeah. One major symptom is called post-exertional malaise, which you have. That means if like we do jogging, like no problem. But those people, if they even take a shower, they will get crashed down for a week or 2 weeks, and to get enough energy to come back to stand up again. And so called post-exertional malaise. So the diagnosis, you have to have this symptom for more than half a year.
Oh,
so
wow.
it's not symptom based right now. So our research goal is trying to identify the objective biomarkers for the diagnosis.
Yeah.
All right. So any other questions before we finish up?
Very good talk. Very, very fascinating. Very important. Very important work.
Someone commented, so instead of Regeneron in the hospitals, maybe you can do vitamin D.
Hey, there are studies that have already been done. If you have sufficient levels of vitamin D, your chances of being intubated are much, much less, if any, compared to people who are deficient or insufficient. But it's not quite that simple. You have to look at both vitamin D and magnesium, because you can't activate the storage form of vitamin D without sufficient levels of magnesium. And what do we find in the American public? 70% of all Americans are either insufficient or deficient in magnesium because they don't eat dark green leafy vegetables. And so we have a real tragedy. I mean, it's that could be easily reversed. So many chronic inflammatory conditions as a result. So if you were to, I was thinking when we leave here, I was going to look up, are there any studies on ME and vitamin D? So if somebody did a study, they got ME and they give them vitamin D and they say, oh, it doesn't work, they probably didn't look at their levels of magnesium. Nothing is quite as simple as it seems. But we really know the tremendous effects it can have on the human body. Imagine it's a neuronal steroid for your brain. It's an immune steroid. All your immune cells have both the enzymes to convert the D3 that you make or take into 2 active forms if you have sufficient levels of magnesium. And what's important to know is that the amount you have in your bloodstream, if you have a chronic inflammatory condition like ME, it would be burned through, it'd be used up very rapidly. You have to keep measuring the blood levels and give a bolus to get it up rapidly. But most physicians, oh, a vitamin, oh, okay, give them 2,000 units. You know? So it's been an uphill battle against the FDA using therapeutic doses of a vitamin because it was mislabeled, and they did studies years ago. How much do you need of vitamin D? Well, the benchmark was the amount of vitamin D you need is so you don't have malformations of your bone called rickets. So then they said, oh, 400 units, that's enough. So that was in multivitamins. It's still in a lot of multivitamins, and that is ridiculously low. So I'm not a medical doctor, so I'm not gonna say how much to take or make. The bottom line is you need to have your blood level measured, and it should be measured once a year at least. But what doctor does that? You know, but it's the most critical steroid in your body. It's more critical than testosterone or estrogen. Every cell in your body has a receptor for it. But Michael warned me that I'd go off on my rant on vitamin D because we studied it in asthma, and I've seen therapeutic doses of vitamin D that I recommended personally totally turn around autoimmune diseases and other conditions. I mean, it's
yeah, because of you, I take 8,000 units a day. But somebody asked, so me personally, I think he's gonna say no, because there isn't a biomarker yet. But Dr. Lee, so somebody can be diagnosed with ME even without prior symptoms?
So one requirement for to diagnose ME is you have to have the unexplained fatigue for more than half a year. This fatigue is not from the cancer treatment, not from any, it's totally unexplained for at least half a year. A few criteria. The other is you have a post-exertional malaise. You can do whatever you used to do, but now you suddenly you cannot do at least for half a year. So the half year is the criteria.
So yeah, I think to the person that asked the question, you do have to have symptoms because as of right now, the current criteria is based on symptoms. Dr. Hartmann, somebody asked, what's better, the liquid form or the cap form if you were to supplement?
With vitamin D, I
Yes.
don't think it would make any difference. But the important thing is get the blood level measured. If you have a chronic inflammatory condition, you can even do it yourself. You can prick your finger. You can go on the web and look at home vitamin D testing. Prick your finger, put a drop on a piece of paper, and you send it off, and they'll measure your blood level, and then you'll know how much you should be taking. Because if you start taking 50,000 units once a day thinking, oh, more is better, you can develop hypercalcemia. And then over time, you could develop ectopic calcifications. That's very rare. I mean, they've given volunteers, I think, something like 500,000 units daily for a month or something crazy. But it can lead to hypercalcemia, confusion, etc. So you want to, but you need, if you monitor the blood level, then if you have a serious inflammatory condition, I had one student at the beginning of the semester, he came up to me and said, oh, my immune system is rejecting my liver. I'm going to need a liver transplant. My doctor said I have autoimmune hepatitis. And I said, well, go to your doctor and ask him to do blood level. His was 0. So he took, with the approval of his physician, 50,000 units once a week. By the end of the semester, his liver enzymes were normal. He didn't need a liver transplant. But if he would have continued in that totally deficient state, because vitamin D as a steroid is needed to produce T regulatory cells. So the other thing that we could measure in these ME patients is whether or not and we can do that in my lab with flow cytometry. We can measure the level of T regulatory cells, which control and dampen the immune system. If you're deficient in D, you can't produce them. You can't make them. That's how critical that is as an immune steroid and natural anti-inflammatory steroid. And unlike prednisone and dexamethasone, it does not cause osteoporosis. But you go to a doctor with an inflammatory condition, he will give you prednisone. If that doesn't work after time, he will give you dexamethasone until you become glucocorticoid resistant and it doesn't work anymore. But chances are he's not going to measure your blood level of D, and he's definitely not going to give you a therapeutic dose of vitamin D3 because it's just a vitamin. You do get all the vitamins you need in your diet, right? But it's not a vitamin. You don't get all you need in a vitamin. You can't get it enough in your diet. You're supposed to make it sunbathing.
Yeah, and for the ME/CFS, what I learned from patients, they said they went to see doctors. The doctor will either try everything, all the blood tests they can do in the hospital. Everything is normal. That's typically what they receive. Everything is normal. All the blood testing, the scan, whatever is normal. So that's the reason in the beginning, years ago, the clinicians thought this is probably just some psychological problem. And the research in my collaborators and include some in my lab, in recent years, we through the genomic sequencing, those analysis, we find the one feature is the immune response is different. The cell, the immune profile is different. The indicating there is something happening by the immune system are involved in this disease. It looks like the patient's in patient's cell, the cell is working hard, very hard. They use up a lot of energy to defend, still defending something. We don't know what, but it's still defending something. But if you do a normal clinic to a hospital, like a physical checking, now everything is just normal.
Yeah, but
Everything-
normally they do CBC and CRP, and they rarely they do vitamin D or magnesium. And I'll tell you one thing they never do. So it's more complicated than I related here. There's one other player besides vitamin D and magnesium, and that is something called vitamin D binding protein. And there are isoforms of vitamin D binding protein. So wow. When I look at that disease, I hear about all this inflammation, I can't help but think that they can't produce T regulatory cells. They don't have an anti-inflammatory steroid. It's sad to see. So we'll definitely get together over lunch and talk some more. Because we have to let these students go study, or they're going to get student study malaise
Yeah, they need to study.
and not be able to get out of their shower.
Dr. Lee and Dr. Hartmann, thank you so much for everything. We appreciate you guys.
Yeah,
Thank you for inviting us so we meet other brilliant scientists. It's wonderful.
Yeah,
Very wonderful.
honor to have the chance to talk with you guys.
And Iman, I'm glad you tuned in. I see your name there.
Yeah, thank you, Dr. Lee and Dr. Hartmann. We like to host this once a year, and we're always honored to have professors like you two just so passionate about your work. So definitely, we're going to invite both of you two for next year's seminar as well, and hopefully we get some good updates, and including myself and Michael, some updates.
Yeah, Klein Levinson and endometriosis and
Absolutely.
onward and upward. And hopefully next time we meet, the Ukrainians will have defeated the Russians. So let's hope for that.
Let's hope.
Yeah.
All right, guys, we'll let everyone go. Once again, Dr. Hartmann, Dr. Lee, thank you so much. I'm pretty sure everyone appreciated it.
Dr. Lee, I'm shaking your hand virtually.
Yeah, virtually, yeah. Nice to meet everybody. And
All right, guys.
I just wanted to mention really quickly before everybody logs off, we are also hosting another event, HOSA and MMA during pre-health week on March 17th, which is a Thursday at 6:30. And we're going to be hosting a physician panel with a pretty broad range of physicians, dentists, PA, podiatrist, pharmacist, chiropractor, physical therapist. So make sure you check us out on the website, the pre-health week website, and keep looking at your email because we'll send out the link for that. But thank you guys for coming. Thank you to our wonderful professors. You guys were awesome. A lot of good conversation happened tonight. Hopefully we can make some breakthroughs and you guys can connect and do more great scientific work. Thank you so much.
okay, thank you.
Very welcome. Very welcome. Our pleasure.
Yeah.
Time for dinner.
Okay, thank you, everyone.
Yep.
Bye-bye.